Velkomin á Málstofu Hafrannsóknastofnunar. Mute. Hvað segirðu? Afsakið þetta. Eins og þið sjáið þá hérna skólinn hjá Sameinuðu þjóðunum í fullu gangi. Afsakið hérna umsátrið. En við lifum það. Í dag ætlar Chris okkar Pampoulie að vera með erindi hérna erindið og loðnu. Mjög áhugavert erindi. Ég ætla að segja hérna í upphafi að ég þarf því miður að fara mjög fljótlega, en það verður hérna tími á eftir til þess að spyrja Chris og endilega nýtið tækifæri og spyrja hann eða viljið. So, switching to English. Chris. Chris did his undergraduate studies in biology and master's degree in oceanography in Montpellier in France. He did a PhD in ecology and evolution at the same university and graduated in 1999. In his PhD, he was investigating the effect of severe floods on the reproductive biology of gobies in lagoon system. It's gobies, right? I think, yeah. He then did one postdoc at the University of Louvain in Belgium, where he studied population genetics for 2 years. Followed that and went to Finland, Helsinki, Finland, where he studied behavioral ecology. He then joined our institute in 2002 as a postdoctoral student working on cod genetics. Christoph's research interests focus on the application of genetic genomic tools to fisheries management and conservation, and his lecture today fits nicely within that. So, Chris, the floor is yours.
Yes. Thank you very much for the presentation, Linus, and good afternoon, everybody. So today I'm going to speak about the use of eDNA to try to assess the distribution and abundance of capelin. It's quite a catchy title, and it's a project that we started in 2018, and that was funded by Rannis Techniþjónustuður. It was a bit delayed. It was supposed to last for 3 years, but it lasted 4 years because of the pandemic. So there are lots of collaborators there, but the project was mainly done between our institutes and Mattis. So before I go in details into the project, I would like to say what is eDNA. Actually, it's fairly easy to explain. As we stand here in the room, we are actually releasing eDNA. Okay, that's environmental DNA. I will call it eDNA. So because we are losing cells from our body, from the skins, from the hair, everywhere. And it's the same with organisms. They are actually releasing constantly cells in the environments they are occurring. So it's the truth for a bird that is scrubbing his back on the tree. It's the truth also for animals that are pooing and peeing, which are most of them, and also swimming in the sea. But there is one thing that I'm not going to do today, is that I'm not going to speak about what this picture is showing, biodiversity. So most of the eDNA studies are about studying biodiversity evolutions, and that's a specific approach. I'm going to speak about a species-targeted approach. So we are only going to look into the DNA of capelin. And yes, I'm not going to talk about a frog, but capelin, yes, just to be sure. The other question that we do really often have is that why are we interested in eDNA? Why eDNA instead of something else? Well, I might remind you that lots of publications have been appearing in the last 10, 15 years suggesting that we are actually facing the 6 mass extinctions of species in the world, and that's the biggest one since humanity. So, and when we think about this, we think, of course, about our impact, human activities, and also human research. It's also including the fact that to study stock assessment, for example, we have to kill many, many, many fish. So there was this brilliant idea that we could use some non-invasive and non-lethal technology maybe to study things. And it's even more alarming when you're actually looking at how many species we do have on Earth, because all the statistics that are based that we are using to assess biodiversity decline are based on the fact that there is only 1.2 million species described at present on Earth. And the estimate is that we could be around 10 to 100 million species on Earth, which means that basically, if you look at those numbers, approximately 86 to 91% of life is not described. So it is quite alarming. And Pierre Tabellet and co. I think it was the first time you mentioned that was 1998, actually. So you see, eDNA took a lot of time to come in the mind of researcher because the first book that was published on this was 2018. But so he was saying and suggesting that in areas that are not really easily accessible, like the Orinoco rivers in Venezuela or in the mesopelagic deep layers in the ocean, we could use other techniques than just trolls or destroying the forest to try to understand what kind of biology is around. And I could show you many, many other examples like deep sea organisms and then the Madagascar stone trees, forests. Well, you know that one. We are not so far from it. So those environments are not really easily studied because they are not that accessible. And the creatures that are living in them are not accessible. So the idea was that actually using environmental DNA, so DNA that is released by organisms when they are living in an area, in an habitat, could be actually collected. And that will help us not to disturb the animals, but it will also help us to see its movement and its presence in the area. So that has been done on carnivore detections in Sweden, actually, with lynx. It has been done with water samples in Australia, where they actually found out that one of the species of birds that they thought was extinct was actually not extinct because they found the DNA in the water. And many other examples like the Olm salamanders in caves in Slovenia. It was thought to be totally extinct and they found traces and then they sent robots in the caves and they found it. So it has many applications, but of course, one of the things you have to remember is that to do environmental DNA, you need to have a sequence for the organisms that you're studying. That's the prime things you need to have. So why capelin? Capelin, because you probably all know this story. This is the distribution of capelin prior to 2002. So capelin is a fish that is actually spawning in the southwestern area of Iceland, which is represented in red there. And the fish actually die after it spawns, and the larvae are actually drifted towards the north of the Iceland, represented in blue there, which is a nursery area. And when they are growing, then they move in the green area, which is the feeding area. And that used to be in the Icelandic Sea, north of Iceland and southern part of Jan Mayen. But all of a sudden, after 2003, this is what happened. The feeding area and the nursery area of the capelin totally changed and shifted towards waters in the East Greenland area. And if you look a little bit further, this actually got consequences onto the fisheries, because if you look at the logbook, and this is a paper we published in 2020, then the left figure is actually showing you the periods before the changes in distribution, and you can see that the fisheries, based on the logbooks, these are the 4 first weeks of January. They are mainly located in the southeast part of the country and really concentrate in the southeast part of the country. But after the changes, then the fisheries got spread much more, and they are going from the north to the southeast of Iceland now. So because of all these changes, we wanted to know if we could actually track or detect capelin using eDNA and see if we could actually, try to estimate its distribution around Icelandic waters. The other objective was to try also to quantify it, not only detect it. So based on a certain concentration of DNA, can you say how many fish are there? And we had planned to do some experiments on board Arni Fredriksson to estimate this. And then we wanted to do some kind of modeling of eDNA distribution using drift model and some kind of ecosystem models that were already published when we started the project. So, and the ultimate goal was really to assess if the eDNA signals reflected detection and abundance of capelin that we were observing with the monitoring acoustic survey. So one of the things that went really wrong in the project already is that the quantification experiment on the boat didn't go as planned, so we couldn't perform it. It totally failed. And so what we did instead was to try to find correlation between the eDNA concentration and the acoustic values directly, without having any experiments where we could confish and see how much DNA they were releasing. So a little bit about the sampling strategy. So we used Niskin's bottles, like you can see there, and this nice, on the right side, you have the pumping system that was developed for it. It's the total right one with the 8 plastic supports. You can actually see a glass system there that we are using to filter eDNA. So, and these are the stations that we did samples in 2019 and 2020 on the East Greenland area, where capelin is during the capelin survey. And the blue triangles that you have there, the reason why they're blue is that because those are stations that we indeed repeated from one year to another one. So it's 2019 and 2020 in the exact same position. And we did 5 depths. So we filter water at 5 different depths, which was surface, 20 meters, 50 meters, 200 meters, and 500 meters, or the deepest part where we could actually get water samples. So in total, we had more than 600 eDNA samples through the water column in the East Greenland area, and we all analyzed them. So let's go a little bit into the tedious part now, the genetic part. So one of the markers that we really use very often for species identification is the cytochrome oxidase subunit 1 from the mitochondrial DNA. It's a gene that is quite conserved from species, but so you can amplify it with kind of universal primers, but you can also design primers so that you will actually amplify the part of the genes that is just from the species you are targeting, and that's what we did. So the first thing you have to do is to, I'm not sure you're going to see that. Is it? Yeah, that's okay. So this is what we call the consensus sequence of the capelin, which means that we amplify this region for 94 different capelin, and then looked at the average sequences. And then you have a typical sequences with some sites that are varying. And you compare these to the 28 species that are below. So the upper part is the capelin one, and the lower part are all the 28 species that we have been adding in the group to see the difference between capelin and the other species. And that's how you design your primers, because you're looking at the area which is different between capelin and the others species that you have in the waters around him. So, and we compared it to cod, mackerel, Atlantic herring, salmon, brown trout, rainbow trout, lots of species, Atlantic and spotted wolffish, redfish, oceanic and deep sea ecotypes of redfish, the Norway redfish, the golden redfish. So most of the species we could think of in the marine environments. Not all of them, because that would have been a lot of work, but at least the most common one. So what we did then is that we developed a framework for the level of detection and the level of quantification of the DNA of capelin. And you can see there is an amplification plot example here where we had samples of capelin with known concentration from 10 nanogram to 0.001 nanogram per milliliters. We actually did those solutions. We took DNA from capelin and we dissolved them at certain concentrations. And each time that we did a run to test the eDNA from water, we had those samples in the plates. So this was constantly run with our samples. And it actually helped us to design a framework that will help us to calculate concentration of eDNA in the water. So you have also 2 examples of stations there that is on the table on the sides, which is purple, the station 414, and orange, the 420. And you can see that we are detecting at 50 meters eDNA of capelin in the purple one, and that's the small curves that is at the end of the graph. And we are detecting capelin in many other areas in the 420. That's why you have lots of orange curves. But you see that we also had tusk, link, mackerel, haddock, and cods in the PCR reaction, and we didn't amplify any of these DNA. And then we had water sample as a blank, just to say, if we put just water, still water there, are you going to detect DNA? And then we don't detect anything, which means that you don't have contamination in the lab or in the process from the boat to the lab. So this has to be tested constantly, because if you have something in the water, then you have a problem. And if you detect species that you're not targeting, then that's also a problem. So the level of detection is just to show you how we do. We actually repeat several times amplification of the standard curves that we have with standard concentration and compare that to what we have in our samples. And then you define area where you will say, okay, above this concentration, then I'm detecting capelin, and above this concentration, I'm able to quantify how many capelins are there. And this is what we did, and we came out with, oh, I went back, sorry. 2 concentrations, so which is the level of detection, that's 3.2 10 to the minus 4 nanogram per microliters, and then the level of quantification is 6 10 to the minus 4 nanogram per microliters. So everything that is lower than 3.3 10 to the minus 4 and that we detect as eDNA, then we don't consider it to be a detection. We consider it to be a trace, but not a real detection. The fish might not be there under this concentration. And then in the quantification, we try not to quantify anything if the concentration is lower than the LOQ. So what we did also during this project is actually do repeats in the lab. So each time that we had the DNA samples, we did 3 PCR to try to amplify the eDNA of capelin. So, and you see that, so sometimes it works really well. So the CT, I didn't say that, but that's a very important columns. It's the cycle thresholds. So it is the number of cycles that you need to do to actually detect eDNA of capelin. And the eDNA-C, it's the eDNA concentration. So we use both of them to actually be able to assess if we are detecting capelin or not. But you see there from 50 to 500 meters in the samples, in the first run, we are actually detecting quite a lot of capelin, and we are also detecting it in the second run and in the third run, which means that at the end you have a quite high concentration of eDNA of capelin. So we do an average. It's not a real value. It's an average of 3 different amplifications that we are doing to assess the concentration of DNA. But you see that, for example, at 20 meters, sometimes we just don't detect anything. There is nothing. And then you see that for the first run, that is run 1. But then for the others, we are detecting something. So it happens that the first PCR don't do anything, don't amplify anything, but that the others are. And we also have to do repeat of stations. So sometime when we have time during the surveys, which is not that often, we need to repeat the stations, just to be sure that if we don't find anything in the first run, we are not finding anything in the second run. And in the ocean, that's actually quite difficult because you have lots of currents and DNA disperse quite quickly. So I'm showing just a very nice example where we have blanks and everything, and where we are actually not detecting anything in the repeats and in the original stations, but we still have trace and no complication of data, because I didn't want to discuss the other one where we actually have the first run detection and then the second run no detection, but we can discuss that later on. So what about results? Enough about the blah, blah, blah. So we did samples 2019 and 2020, like I said, and here you have an acoustic signal grading figures, which is showing you where the capelin was in 2019 based on the survey. And here is the eDNA results. So that's the eDNA results with everything. Everything we detected, every single concentration, without level of detection into account or anything. But if you actually look at the level of detection, then this is what we have. This is where we are actually really detecting capelin. The rest is just trace or contamination in our samples. That could be one solution. So we did the same thing in 2020, where we had many more capelin than in 2019. I forgot to say that, but in 2019 we didn't have that many capelin in the water. And there in the eDNA, we also detected quite a lot of eDNA of capelin. And this is just all the water column. It's not 0 meters, 20 meters, 50 meters, 200 meters, 500 meters. It's just all the water column that we combine together to see if the horizontal representation of this could actually reflect the same thing as the acoustic. Of course, some of those dots in the eDNA are due to detection at 20 meters, some others are 200 meters, and some other at 50 meters. And these are actually results that are under revision right now in eDNA journals, and that's actually what one of the referees asked us. They asked us to separate the different layers of the oceans to see where the capelin was really compared to the acoustic. I will come back later on to this. But again, if you look at level of detection, this is actually what we are detecting as real presence of capelin. The rest is just probably traces of capelin due to currents or due to the presence in previous days. Another thing that we try to do is that, I don't know if you remember those blue triangles that I showed you at the beginning on the map of sampling. So those are two transects that we repeated from 2019 and 2020. So here's the results that we have for 2019 on transect at 72 north degrees and 70 degrees north also. So you can see the results are different for the eDNA in the transects. But the interesting part was when you start to compare this with acoustic signals in the same transects. And you see that, okay, if you try to report the acoustic signals that we have on the right side to the eDNA graph, it's kind of fitting a little bit, but not perfectly. And you see that you also have, because it's a graph that is actually interpolating quite a lot the eDNA data. So that's not really true data. That you have detection in area where you actually don't have capelin. And this is one of the problems that we have been having, is that we actually didn't sample water enough on transects to be able to say anything about what is happening in the dispersing and sinking rate of eDNA. So you see for the 70 degree north also that where we are detecting capelin, we have very low eDNA detection levels there. But we have a very nice one at this point, the square one, and then there we actually don't have any capelin if we look at the acoustics. So the nice thing is that you have 72 degree north and 70 degree north just on the same slide, because when you have nothing in this square on the 70 degree north, you are wondering, you know, is it not some kind of dispersing from the currents from the 72 degree north? Because actually the currents here will be just north-south. So there is a possibility that actually we are not detecting eDNA in the right areas in some of those transects simply because of the currents, that the fish was higher up and the DNA was dispersed by the currents to the next station. So what we are basically thinking of is that to be really able to understand all those things, we actually need to use the eDNA data that we have been collecting now to do drift models and sinking rates models. So, and I say anybody interested, well, there are already people working on this in the house, so I hope that this will be solved soon. So I could show you the same graphs for 2020, and here is an example again of the same transect. So you see that from one year to another one, the detection of eDNA and the acoustic signals are changing quite a lot. That's quite normal. You don't expect that to be stable from years to years. But again, this time you see a little bit more, you see a better correlation between the eDNA concentration and the acoustic signal, at least in the upper part for the 72 degrees north transect, but not for the lower part. Again, you can see that you have lots of capelin detected with the acoustic in the surface, but at 70 degrees north, but then we don't detect any eDNA there. Yeah, that's the one that corresponds. Yoo-hoo. But another way that we have been asked to present those things based on the revision is this one. And this is just an example that we quickly did with my co-authors. And because referees were asking us actually, how is the eDNA detected compared to water masses? And then one way to look at the water masses is the salinity and temperature. So there you have the temperature profiles on this transect 72 degrees north in 2020. And then you have the salinity profile on the right side. You see also the bottom, that's the white part, that's the bottom of the sea. The blue dots are actually the acoustic signals where we detect capelin, and then the other dots are actually samples of eDNA. And the biggest concentration that we are getting is these yellow dots and these small gray dots. They're really close to the acoustic signals of capelin. The rest is actually just traces again, and it's not considered to be detections. So representing it like this is a little bit more conceptual. We see more what is happening compared to this kind of graph. So that is what we are actually doing right now. So the next step that we did was to try to apply eDNA occupancy models to our data that are usually based on repeat station, repeat depths and everything, but we managed to rig our data to fit them into the model. And the first things we saw, and that we were quite happy about it, is that you actually have a correlation between the eDNA concentration and the NASC value, so the acoustic value of the capelin. Even if it's not totally in the same spot, if you take the closest spot of acoustic signals, then you realize that there is a correlation between your concentration of eDNA and capelin. And then we played with environmental variables just to see if we could see a pattern there. And we took like salinity, temperature, and velocity, current velocity. And the first thing you see, it's the graph A. Then you have the more current speed you have, the more eDNA you detect. And for the salinity, that's actually the reverse. The eDNA is not detected above 32 PSU in salinity. So there is a decrease from 32 to above. There is a decrease on eDNA concentration. And then the last one is temperature also. After 2 degrees, there is a trend in decreasing concentration of eDNA with temperature. So this can be due to many things. It can be due to well, current speed. We know, if the fish is actually present in very rapid water masses, then you are likely to find lots of DNA there. But salinity and temperature can be due to different things also. That could be characteristic of the currents, but it could also be that at higher temperature, there is a higher metabolism, and then the eDNA is actually used by other organisms as an energy sources, like I'm thinking about bacteria and all those things. But then I will say, we faced lots of challenge during this project, many challenge. It was not that easy to collect the samples. It was difficult to organize, and it was not a eDNA monitoring. It was done during the capelin cruises, which means that there's lots of things that we couldn't do. And one of them was the eDNA production and shedding rates of the capelin. We actually don't know how much the capelin is releasing when it's leaving in the water. So that's a very difficult things to imagine. And you see that from one species to another one, this rate is actually changing. We know that, even in the marine environments. Some species actually shed more DNA than others in the environment. So you will detect those species much more easily than others, basically. And then the other problem we had, it was decay rate. We actually didn't know at all how long the eDNA of capelin was staying at one station and how long it was staying in the environment. So you see here, I'm giving you 2 examples, the one of European flounders and sticklebacks, and this is done by experiment, and that is what we were supposed to do with the experiments, but we couldn't. It's just to take a number of fish, put them in a tank, release them, and see how long the DNA was staying there. So you can see that for the flounders, it's approximately 7 days, but for the sticklebacks, it could be less than 1 day. From the repeats where we actually found DNA of capelin, so we do a station, we do eDNA study, and we come back an hour and a half later. So in the first time we detect eDNA, in the second one we are not detecting eDNA. So that can be an information that, probably not that it's degraded in 1 hour and a half, but that it's gone due to currents, So there are lots of parameters that we don't control there, and then we need to investigate a little bit more. So like I said, to continue, well, we need to basically work on this decay and drifting and sinking rates in the future if we want to continue to work on eDNA with capelin. The framework is there, but there is more to do. But still, it's not that bad. There is a nice correlation between the eDNA concentration we see, at least on the horizontal levels, and the distribution and the range of the species. These correspond quite well. It's getting much more complicated when we are starting to add the third dimension, the depth, because then we really need to have a dedicated monitoring survey to understand what is happening, and if we want to see the impact of the water masses, which I think is the most important thing to study when you're doing eDNA, specifically in the open ocean, because the currents there are quite important. And if you want to summarize a little bit the eDNA occupancy model results, you can probably pinpoint to the fact that this is pointing out to the East Greenland currents, all indication that you find more eDNA of capelin along the East Greenlandic current. Yeah, and so like I said, this is a review paper now in Environmental DNA Journal. Thank you very much for your attention. Do you have any question?
I can speak very loudly.
No, just take the microphone. You need to have the microphone. Sorry.
Hi, thank you. Really nice presentation. Impressive work. It's just a technical question. the quantity of DNA. Well, what's the unit? How do you quantify? Is it how many reads of DNA you get, or?
No, that's concentration directly.
Yeah, concentration
it's based on the
directly.
CT cycle of the qPCR.
Oh, qPCR.
Yeah.
All right. Thank you.
No reads.
No reads.
Yeah.
So no PCR prior to the qPCR.
No, directly quantification. Yeah.
Nice.
No more question?
Thank you for your presentation. What I want to ask is, you tend to find DNA which has drifted with the current, but not at the spot where the capelin is. So my question is, are the capelins not extruding DNA continuously?
Yeah, they are, but I guess it's probably, we don't know actually. I don't know. I'm nobody has done that work before, so I'm not sure. They should be. I mean, all of us are doing that more or less at different rates. I guess it really depends on the biology of the species and the currents around and everything. I mean, I don't really have an answer to that, honestly. I mean, I'm quite surprised that that for some of those stations we had capelin around and we didn't find DNA. And the main reason is that we had specific targeted school samples. Like, they send the CTD in the center of the school. And then we did that 4 times, and 4 times we got DNA in high concentration. And for some of them that we did during the transects, then we didn't get it. So I have a hard time to understand this, basically. But it could be due to so many things. It's. I know that we are not the only one to have this problem. I mean, I know people who have been basically taking water samples in pods of killer wells, and they were never able to identify any eDNA from killer wells in the water samples. They found lots of things, but no killer wells. So it could be due to different things. It could be due to the fact that your genetic technique is not perfect. That can happen. And that there are inhibitors in PCR also. We had problems with inhibitors. We had to remove quite a lot of inhibitors. But sometimes it might not work and you don't know it. It's just random things also. That happens.
Yeah, thank you, Chris. I will have to digest what you said, but I'm wondering if you could give your opinion on why there are so much variability in the decay of DNA. These are fish, and it's not so different, which you showed as an example. But have you any ideas?
I think it is a little bit. I mean, if you go back to DNA, not the eDNA, to the old DNA, you can say it's fish also, and I've been working on fish for 20 years, I think, but you still have species for which it's very difficult to extract DNA from tissues. One of those fish, for example, is herring. It's an horrible fish. It's really hard to get good quality DNA from herring when you have tissue samples because of the fat. And it's actually the same for capelin. If you do DNA, it's very hard to get DNA. So I mean, there is some kind of biological things happening on those species, and that's why the decay rate is different. And also the skin, the roughness of the skin, and and the scales, or all those kind of things that could be very different from species to species. That's why the decay rate and also I forgot the name now. The emission rate of DNA of those species is different. So you don't lose cells at the same speed if you are an anchovy or if you are a flounder. And then there is age also is another problem. When you have fish older that are almost, I'm quite sure that if you were to take eDNA samples during the spawning time of capelin, then you will have a higher concentration because they are investing a lot in reproductive efforts. So they are probably losing quite a lot of cells at that moment. There are lots of factors, it's and we almost know none of them. So we know a little bit of some of them, but not all of them. But I would say it's proper biology that is actually explaining those rates. Yeah. Anything else? Okay. So thank you very much.